Alright, so in today's lesson, we're going to keep talking about proteins, but we're going to talk about the 4 levels of structure of a protein. And this is really important because we love protein structures, but mostly it's important because the structure of a protein gives it its function. Alright, we're still in Nature of Biology, page 50 to 56, and I'll give you those Nelson biology books. If I haven't, please remind me. Pause the video here and have a go at these 3 quick questions. Alright, so in this first question here, we're seeing the diagram below represents 3 of the major macromolecule groups living things. Gosh, wow. We don't study all of these anymore. We study this group here, which hopefully you can see is a phospholipid. Alright, and we certainly, from year 10, you'd know as DNA. Alright, this last group here, we talk about it a lot, but we don't study it structurally, but that's just a carbohydrate. Okay, so synthesis of macromolecules from these groups involves, so we have energy generating condensation reactions requiring the input of water molecules. Okay, so energy generating reaction requiring an input of carbon dioxide molecules. Let's ditch that one. We know nothing's going to require an input of carbon dioxide molecules. Energy requiring condensation reactions requiring an output of molecules, or energy requiring reactions with an output of oxygen molecule. Let's ditch that one. Okay, so that was really quick way to ditch those 2. So are we getting an input or an output of water molecules when we are synthesizing? So we are putting these molecules together. Hopefully you know that we have an output of water molecules. So it must be C. Let's check the first part. Do we require energy? Yes, we do. Alright, so question 5 said, if a sample of macromolecule from group C were chemically analysed, you expect to find that it contains fatty acids and phosphate. Phospho is the phosphate, lipid is fatty acid. Cool. Cool. We're going to have a look at this examiner note in class together. Alright, so in the study design, we've got nucleic acids and proteins still. We're looking at their functional diversity of proteins and the nature of the proteome, so that was mostly last class. But we want now to talk about the functional importance of the 4 hierarchical levels, so they're like ranked levels of protein structures. And we're going to go through that polypeptide chain again from an amino acid monomer by this condensation polymerization. And there it is. It's a much nicer picture than Mr. Mello drew last time, right? But what we're having, remember, we've got this side here of the, this side here of the amino acid. So here's the central carbon. So that's an amino acid in blue here. That's a carboxyl side. And we've got this side here of this green amino acid is the amine side, okay? And so the carboxyl side loses this OH, the amine loses this H, all right? And what we get is a water molecule forming, all right? And we get this bond forming called a peptide bond. Hopefully remember that from last class. One important thing that the examiners actually want you guys to have, which isn't represented here, is that we require an input of energy into this reaction. Okay? It requires energy. Okay? So, by doing that, we're forming what we call the primary structure of proteins, all right? And what the primary structure of proteins is, it's just simply the linear sequence, so if you were to imagine one after the other, of these specific amino acids bonded through peptide bonds. And this is all coded by specific genes inside the DNA, okay? Really important, peptide bonds are extremely strong, difficult to break covalent bonds, okay? Once they're formed, it's going to be really hard for these kind of bonds to break, and this is going to be really important later on. All right? pause the video here and grab the amino acid starter kit because we're going to do something with that, okay? Alright, now that you've built your primary structure, so your linear sequence of amino acids, we have the secondary structure of proteins, okay? So that linear sequence of amino acids, we've got, off those off the carboxyl and the amine groups, we've got lots of other hydrogens there, and we get some hydrogen bonds forming in between these groups, okay? And they form folding and twisting the chains, these polypeptide chains, and we get what we call the alpha helices, the beta-plated sheets, and we get some random coils. These are just random things that happen. Okay, so for example, here we've got an alpha helix, so you can see it sort of looks like a twirl like this. Here we've got a beta-plated sheet, which just means that it's a sheet, and then it goes back, and then back, and then back, and then back, and here we've got a random coil, so a mess. All right, those alpha helices, beta-plated sheets, they're the really important ones. Here's the molecular view of an alpha helix. Here's the molecular view of a beta-plated sheet. All right, now grab your amino acid starter kit again, all right, and we're going to want to make an alpha helix, a beta-plated sheet, and a few random coils into amino acid polypeptide chain. Really important, we're keeping that structure 2D still, so it's flat. It's not a 3D structure yet. All right, now we've got that secondary structure. What's going to come next? The tertiary structure. Luckily, the naming of this is pretty straightforward. All right, what the tertiary structure is interactions between those R groups, so those groups that change all the time, that cause the protein to fold into now what's a three-dimensional shape, okay? It's the tertiary structure of a protein that determines the function of the protein. All right, and here's a few examples of these bonds forming. Okay, so we have here an ionic bond forming between a positively charged R group and a negatively charged R group. We have a disulfide bridge forming between R groups with sulfur groups on them. Right? We've got a hydrophobic interaction. So these two groups, they're both hydrophobic. They're scared of water. So they're going to come in and attract each other inside of that protein. We can have simple hydrogen bonds forming. Okay, so lots and lots of things can happen, and things that look like this here, for example, is a channel protein, a channel protein that's in the membrane of a certain cell, okay? So here we've got lots and lots of beta-pleated sheets, and they're forming this little cone structure so that molecules can go in and out of a cell, all right? Or we can have lots and lots of these alpha helices interacting to give a specific shape to this specific protein, giving this specific protein a special function. All right, grab your amino acid starter kit and call me over, because we're going to make some interactions between these R groups, okay? This time making a 3D structured polypeptide. All right, the final structure that some proteins have, not all of them, but some protein have, is what we call the quaternary structure. And this is what happens when we get two or more, or more polypeptide chains forming together, all right? They both form a tertiary structure, and they join together, okay? I think the best example of this is hemoglobin. Hemoglobin, if you don't know, is the protein that lives inside of red blood cells, and here it is, a little picture of it. And what it does, it's got this, in light blue here is one tertiary structure, polypeptide chain, and green is another one, yellow is another one, and pink is the last one. And they all interact, those R groups of each of those interact with each other, forming this big jumbled protein. And then it has a specific shape here in the middle that beautifully fits a couple of oxygen molecules. Those oxygen molecules get carried by hemoglobin, and it means you get to breathe. Okay, we're going to grab the amino acid starter kit and join two together. Okay, now that you're done with the amino acid starter kit, let's have a go at watching this quick video in class.